So I already went through this argument about RNAs. What do people imagine when they think of an RNA world? There are really kind of two extremes for this. And I talked about this briefly when I talked about viruses, right? One is the simple RNA world hypothesis. So it turns out you can make really rudimentary RNA replicases. These are RNAs that have the polymerase activity and will bind a nucleotide and in a templated fashion will polymerize it to the end of the RNA chain. You can go a couple of dozen nucleotides this way. You can do a slipping thing. So an RNA, and these are synthetic molecules. So RNA replicases are certainly within the realm of possibilities. What you would imagine is a little bit like a virus replication, where you have a negative and positive strand. And so the replicase would replicate in a templated way, would copy a copy of itself on the opposite strand, and then that would be the genome, and then another replicase would replicate that to create the replicase strand again. So it would go back and forth between genome and replicase in the same way that a lot of viruses go back from positive to negative strand during the replication. The difference is the replicase is an RNA rather than a protein replicase, and it's actually one of the two strands. You can imagine then that one strand could serve as a template for the creation of proteins in some way, and probably in a fairly indirect way, messenger RNA with some tRNAs, and the ribosome wouldn't necessarily catalyze it, or at least not a complete one. And so you have an RNA translator that makes proteins. And these would be very simple proteins that in a very nonspecific way would facilitate certain kinds of reactions. All you have to do then is to replace this genome, RNA genome strand with a DNA, and you have what amounts to today's world, right? You have a DNA genome, an RNA translator that makes proteins, a protein replicase that makes the genome. And so in this scenario, the RNA world was a very transient place. A lot of people believe in a complex RNA world. An RNA world with essentially all the metabolism we have today, catalyzed by protein enzymes rather than with RNA enzymes rather than protein enzymes. The evidence they have for this is that an awful lot of the biochemistry we use today in biology is carried out by nucleotide coenzymes. ATP, GTP, NAD, FMN, the list goes on and on and on. These are all nucleotides. These folks would argue that those nucleotide cofactors are the last remaining relic of the original RNA enzyme, and that the protein has taken over all the other function except for the essential catalytic function of the enzyme. No, I don't think that. The reality, though, is we don't have a lot of information. I think that at this point, you just have to use your best judgment. I don't think you can definitively say anything at all. And that brings us to this reality check. There are some big problems to the RNA world hypothesis in general. I think the RNA world hypothesis is the best we have. Someone reported to me that the folks in biochemistry think I don't believe in the RNA world. That's not true. But I think there are fundamental things about the RNA world hypothesis that we don't have the answer to yet. And here's a big one. Activated nucleotides. You can't make an ocean or a pond out of GTP, ATP, CTP, and UTP to serve as substrate for all the stuff going on. It doesn't happen. The nucleotides are pretty good. The bases themselves, D-ribose is a nightmare. There are hundreds of pentose isomers. And if you synthesize ribose abiotically, you get them all. How do you fish the right isomers out to make RNA out of? Because every one of those other ones, L-ribose, for example, is going to be an inhibitor of your enzymatic system. And there's no abiotic way to make ribose specifically. That's in contrast to adenine. Adenine you make out of cyanide pretty easily. Guanine, not so bad either. Pyrimidines are a little harder, but ribose is a nightmare. RNA is very unstable. Any of you who worked with RNA in a lab before know this. Now, a lot of this is rumor and I don't know, paranoia in some way. A lot of it is because people throw RNases around like it's candy, and that's a problem. But it's absolutely true that RNA is very highly susceptible to metal-catalyzed base hydrolysis. And so if you throw a little bit of metal in, like lead, and even manganese and zinc, and the pH goes up a little bit, that 2'-OH is a prime target for hydrolysis. And at high pH, at low pHs, you get depurination. So RNA is pretty pH sensitive, and certainly that's a temperature thing. And so there's a question of how do you protect an RNA in an abiotic world? I mean, certainly if you throw RNA out into a sterile environment, it doesn't last very long. Do I go on in this conversation? So those are all problems with the RNA world. Let's go back a little further. I don't think these are insurmountable. I think the next thing we talk about will help with some of that.